Nice to meet you again. I think most of you are in the genetic group, right? Or in the section of the training, right? Okay. Are we waiting for Asif first or should we start now? Okay. Okay. Last week I talked about a SNP, if you remember. That is about the genotyping, right? For the last week, we learned about genome assembly, genome annotation. So from the genome annotation, you know the location of the gene on the genome, right? So from the annotation, you know the position of the gene. If you do another experiment called the RNA sequencing, you will sequence sequence the molecule of the mRNA. From the molecule of the mRNA, you can map back to the genome that has the annotated region of the gene. So you can count number of the mRNA at each gene location on the genome. So from the number of the mRNA, you can find the extraction of the gene of that gene from the annotation, and that technique is called the RNA sequencing or RNA-seq. And there are some problem. Actually, not problem. It's some limitation in the RNA sequencing. So in some case, you need to do the validation. And normally, people usually use the RT-PCR or quantitative PCR to count the number of the mRNA for the validation. And another technique called the nanostring. So the nanostring also count the number of the mRNA molecule. Okay. So another technique to count the number of RNA is nanostring techniques. So today I'm going to talk about how to use the nanostring to count the mRNA for a non-model organism. In this case, it's a non-model plant. So actually, we use the nanostring to study the response of sugarcane. The response of the sugarcane to drought. So because the drought can cause up to 60% of the yield loss, so we need to know what gene of the sugarcane that are expressed when it the plant experience drought stress. So we consider to use the RNA sequencing, or RNA-seq. So we have to set two conditions. In the first condition, actually there are three. There are another one called the control. So this is the first genotype, and this is the second genotype, or the first cultivar and the second cultivar here. And in this case, we stop watering, so stop feeding the water. So the plant experiences drought stress here, and for some time we cut the sample from we cut the leaf from the plants. So we extract the RNA sequence. We extract the mRNA from this leaf, and then do the RNA sequencing. So the RNA here means that the mRNA. From here, we can count the number of mRNA and do the extraction analysis. In the extraction analysis, you just compare the number of the mRNA between two conditions. This is the control condition, like the plant that receive the normal water condition, and another one for the plant that experience the drought stress. So from here, You can count the number of fractions and compare the expression. Then you got the heat map like this. The heat map shows a number of genes here, a lot of genes that can be classified based on the function of those genes. The red color here means that this group of genes, the expression compared to the normal condition, the expression significantly changed. In this case, you see red for the positive number. It means that this gene is upregulated. These two genes are upregulated under drought stress, so increase the expression compared to the baseline. Another case, like this gene has the blue color, so represent the negative negative pole change. So it means that these genes are downregulated in the plant under the treatment, under the water treatment here. So this is some example of the result that you can get from the RNA sequencing. But, okay, sorry, this is another picture that show you about how that we summarize about the gene that change the expression under the drought stress. So we can find that some specific pathway in the plant are changed when the plant receive the drought stress, are treated with the drought stress. So we can further analyze the function of this gene in relation to the water metabolism, I mean the metabolism of plant in response to the, let's say, the treatment of the water. But today the focus is not the RNA-seq. We will show you about the nanostring technology that can be used to validate the RNA-seq result. And I first show you about the video how the nanostring works first using this video. So in this case, it's nanostring normally used to study the disease in human. And from the previous, from this, sorry, I go to the YouTube. From here, you see that when you study the RNA, when you study the gene expression analysis, you need at least two conditions to compare the expression level. And in this case, you need to study the cell inside like a cancer cell here compared to the normal cell. So remember, if you do the RNA analysis, mRNA analysis, or expression analysis, you need two conditions for the comparison. So here is the molecule of the mRNA, right? You see the single strand. And in the traditional technique, yes, in the traditional technique like RNA sequencing I showed you before, from the starting molecule, from the sample, you have to do the reverse transcription to get the cDNA. And after that, because you need a high level of the molecule, I mean, you need more molecule, so you need the amplification step. So this can affect the correctness of the analysis, because in the expression analysis, you have to compare between two conditions. And this step can cause like an unfair comparison, because you cannot control the expression of each molecule. For example, if this gene has a longer length than this one, so the number of this gene should be higher than, in this case, it will be the blue and green, right? Blue one may be shorter than the green one. So the reverse transcription or the amplification of this one should be higher than this one. Then you got the high number of the blue copy than the green copy. This is the like a limitation of the traditional technique. Yes. So this is the real, the number of mRNA from the sample, the original one. I mean, what actually in the sample, the number of RNA in the original sample. But after you do the PCR, the amplification, the number of this one can be varied from this one. So it changed the number. So because of this one, in our study, we consider to use the nanostring to validate our result. Because nanostring, go back a little bit. From the nanostring, from the single molecule of the mRNA, you have like a capture probe, and then another one called the reporter probe. So from this, you see that you can directly count the number of the mRNA molecule. No need for the amplification. So you can reduce the limitation of the traditional technique. And then when you do the purification, select only the molecule that have the probe. This one is called probe, that you can detect color. So it's bind to the chip, immobilize on the chip. And then you can count the number of the RNA. So today I have like a two examples of the chip that we use in our lab to do the nanostring. So I'll show you later. So I'm not sure you can see it or not. It's in the middle here is the where we load the sample in, and then the sample will mix with some probe here or over here, and then flow to another side of the plate here or of the cartridge, and this image will count over here. So you can see this one. This is, I think, so yes, you may get more idea about how nanostring cartridge we will do in the lab from that one. And then from the area, reporter area, you can count the number of molecules like this in this one. Yes, you can do the nanostring with various type of the molecule. Like you can do for the mRNA or microRNA or do the protein here. Because you may have some receptor for the protein here. This receptor had the nucleotide region that can bind to the reporter probe here, and also can detect the fragment of the DNA molecule. But in our lab, we usually do the nanostring with the mRNA molecule. And this is the machine. I think yesterday you have visited our lab in 6th floor, right? You see the nanostring machine in the sequencer room. Okay. Yes, that is about the overview of how the nanostring work and why we use this technique to study, to validate the RNA-seq result. And for this one for the overview, and this figures show you about how to decide the probe. Not to decide the probe, what information that you need to decide the probe. Because in human, actually nanostring have like a code set that's specific to human gene. But in our case, we study in sugarcane. So in sugarcane, then we have to decide the probe. We have to decide this one, the probe. Yes, we have to decide the probe in the green color here with specific to our gene. So to decide this probe, we have first to know what gene we need to study in the nanostring. And the gene got from the RNA-seq result. So we find the gene like this gene and this gene that had the very, very high flow of the down regulation and up regulation here. You got the gene that have different high amplitude of the expression change by considering the four change that you can compare from the control, compare between the control and the treatment condition. So first, you have to know the target gene. The second, you need to know about the information about gene structure. Because some gene, if you have the probe in this area, you will cover the intron, right? So you might not get the number of gene, you might not get this copy of this region from the mRNA sample because this is the intron, right? So we may consider to have the probe that can bind to the exon like this. After that, once you got the probe that's specific to this exon, you may need to do the checking. So the checking is very simple by just find the complementary strand of this region and this region because the probe will bind to these two regions, right? And you have also checked the quality of the RNA because it will affect the performance of the nanostring. So once you extract the RNA from the sample, you may check like I don't know, I never do the lab by myself, but you may use like the concentration of the RNA, for example. I'm not quite sure for the lab technique. Then you check all this. You can use this information to design the probe and do the experiment, do the nanostring experiment as I show you in the slide, in the video. Discover the step of the nanostring run. Okay, so once you got the data from the nanostring machine, you can do the analysis. There are two main important steps in the nanostring analysis. The first one is the import of the data. So actually you got the data from the nanostring machine. There are two types of the data. The first one called the receptor library file, which is specific to the code set. Because you have this one. There are many. This one you can load only 12 samples for each cartridge, right? So it means that if you have 24, you need two, right? You need two of this, so it means you need to run. But these two run rely on the same code set. So you need to have this one to link between these two run with the code set. So you need the RLF files. And another one is called the reporter code count, which is the result from each run. So the result will be the image file that get from this region on the reporter region of the cartridge. So you need to file here and put this into the program called Ensolver program here. And I show you the step to do the data import. Yes. In the software, right, I show you, you need two type of file, the RF file and the RCC file. You first have to load the RF file first because it will link to the code set and will identify the the probe of the gene which you run. Once you upload the RF file, the next step, you can upload the RCC file. So in this case, they have only 12 samples. So it means that they can use only one cartridge to run. So make sure that this code file link to the code set. Yeah, this is can check more about to check the quality of the file, quality of the read count. I show you about later about the comment on this one. Okay, skip this one, not one. Okay, so once you upload the data, to the software, the next thing that you have to take care about is the quality of the data, because when you upload the sample onto this one, the quality of the data will depend on how the camera detect the signal from the cartridge. So the software here report the quality flag. So one of the most important quality flag is about the binding density flag, because this will depend on amount of the RNA in the solution that you upload to the cartridge. Like in this case, there are too much RNA molecule in the sample that you upload to the cartridge. So the software, the machine cannot separate these two probes. So it just actually there are two probe molecule over here, but the software can count only one, because the signal will be mixed together in this region. So in some time, if you increase the concentration of the RNA in your sample, it will cause the negative effect on the nanostring run. So normally we use about 10 nanogram of the RNA for each run, for each sample. So in some case, you have a more number of the target genes. So it means that you have also a lot of probe molecule in this region. So the software cannot, the machine cannot separate two probe molecule. Or the accretion level of the molecule of the mRNA, if you have the high accretion level, it means that you have a high concentration of the mRNA molecule. So it's increased the binding density in this region, and the machine cannot separate the two. And last one, for the RNA integrity. So if you have some damaged mRNA molecule, the probe cannot bind tightly to that molecule because they are damaged, right? As soon as you have the intact one, have like a 16, 15 base, but for the damaged one has only half. So the binding between the probe and the target molecule will be not tightly enough, so it can wash out in the washing steps. So this the integrity and integrity here will reduce the signal, so you will not get the correct number of count. And after you do the data import, the next step you can do the analysis. So there are 4 main steps in the data analysis. The first one is called the sample annotation, that we will, because as I mentioned, when you do the accretion analysis, you need like treatment and the control condition, right? So in the sample analysis, sample annotation here, we'll annotate the sample which one from the treatment condition and which one from the control condition. And you do the background correction because you may need to do the nanostring from two different runs, right? So there are some variation between two runs. And the next one will be the normalization because from you may have the sample from different cells. So from different cells, there should be some variation in the expression level. And this one will help you normalize the variation in the expression level because you have the positive control, and this one will analyze the variation between sample by using the housekeeping gene, the expression of the housekeeping gene for the normalization. And the last one, you can calculate the ratio. So the ratio is from this calculation, you will get like which gene are upregulated and which gene are downregulated. Okay, so this is the overview. I show you the slide. The video again. Yes, from this you got the data upload from the previous video, right? And from this one, you need to do. This is set up the experiment in the EnSolver software. So remember to link with the RF file, select the gene, the sample in your analysis. Yes, this is one of the important steps. So you do from the data, you can do the data sample annotation by using this tab. You got the new column here, and then just name it to the new annotation. And you get some information or some data from your study, like this one should be the control one, and this one should be the treatment one. I mean the treatment group. So that add to this table, add those information into the table like this. So now you know that this group of sample, but this group of sample are under treatment condition, and this group of sample sit on the top here are under normal condition or control condition. So you have to take care about this step because we're gonna compare the expression of this group with the expression of this group. Next, you can do the negative control negative control normalization by removing some noise on the cartridge, because in some case the negative control it means that you should not count this gene. I mean the negative gene should not be expressed, should not be this gene should not be present in the cartridge, right? But if you see the expression, I mean, if you can count the number of this negative control, so it means that you have some error. So you have to subtract some error from the noise that can occur during the imaging when the machine count the molecule. Yes, use the negative control here. Okay. These are another important step in the nanostring normalization. you have to do the normalization with the positive control. So you can see the positive control here, it has different level of the equation or the number of the molecule of the negative gene here. Sorry, the positive gene here. The positive gene, the probe for the positive gene are included in the reagent from the nanostring. So every nanostring reagent has this group of genes. So this gene have different expression level from high to low here. This will normalization the difference between cartridge, okay, or difference between group of the reagent from this tube of the reagent. And another group of the normalization about the normalization with the expression of the housekeeping gene. The expression of this group should be stable among sample. I mean that the sample A, B, C should have stable level of this expression. And actually, Nanostring recommend to use the percent of the CSV here in this about in this range from 0 to 90 here. Sorry, from 100 to 90 here. So if you have like this one, 74 here, you may exclude this one from the normalization. Okay, the recommend value about 100 to 90. So that is about it. The stat. Actually, the information about this one, you can get more detail from the Ensolver manual. Okay. Now after you do the normalization, the next step you can compare the ratio. So when you need to compare ratio, remember we have two treatment condition, the control condition and treatment condition here. And in this step show you the, the point here show you different way to do the comparison. And this case, the video show that it will compare anything with the treatment condition, which is the in this case the treatment was it will compare everything with the unstimulated condition or the control condition. So you have to first select this one, the treatment from the column name here, and select the value, which is the unstimulated condition or the control condition. So if you have stimulate 1, stimulate 2, 3, 4, 5 here, all will be compared with this control. This has only one condition for the example. So it means that if you compare this one with this one, compare the expression level. And this will confirm the run, and then you start the analysis. Check here to let the program do the ratio analysis. And you got the result. There are 4 types of the result after you do the experiment. The first one is the raw data. The first result will be the normalization result. The second one is the group of data. So it provides you about the metadata about the sample, because you have 2 groups of the sample. And you got the ratio data. So we will focus on this one, the ratio data. Okay, so for the ratio data, I show in my slide over here. So In the ratio test result, we compare the treatment with the control to get the full change. So it says that is this the control, how much the expression of the sample under treatment compared to this baseline, up region or bottom region, up or down. So it's called the full change. And it provides you some statistical significant test value over here. So this is the result when you do the ratio test with the nanostring. You got the number of the molecule of the sample in the control condition here, unstimulated sample. And because we have how many copy? About 6 copy in each condition, right? So you got some variation value, or some standard variation value here based on the average mean here. Based on the average value here. So similarly, you got the data from the treatment sample under the treatment condition and standard deviation. So now you have the mean value, you have the standard deviation, you can calculate like a p-value or the adjust p-value. So from here, you got the number for this gene. I did not show the name of this gene, but from here, you got the full chain. So in this case, got the full chain of 3.7. So it means that the expression of the control condition is about 3 times or 4 times higher than the expression of the sample in the treatment condition here. And you got the p-value, but in some case you have a lot of number of the sample. You need the adjust p-value here is about 0. So we say that this gene are significantly, because we focus on this gene, right? The gene under control condition. So we say that this gene is down-regulated compared to the same gene under the control condition. Another one, conversely, you see that this one got the negative value, and the number will be negative 8.5. So the expression change of this gene is stronger than this gene because the number of fold change is greater than this one, right? And it's significantly changed because you got FDR lower than 0.05. So this is how we interpret the result from the ratio test from the nanostring. And yes, this is about the overview that I have shown you before. So in this case, in case of the sugarcane in our study, we have the condition of time. We vary the condition of time. I mean that stop watering for 0 hour, 5 hour, and 10 hour, and see what are changed with the time series treatment. And these are our results from the sugarcane test. You see the blue bar here, the blue stack. The blue bar here represent the fold chain from the RNA-seq, and the orange show the value from the nanostring study. So you can see that the expression of this gene are correlated between the result from the RNA-seq analysis and from the nanostring analysis. So we can confirm that our study is valid enough to conclude that this gene are changed under the water treatment condition. Okay. So in summary, so we in the sugarcane study, we first use RNA-seq to screen for the responsive gene because the RNA-seq rely on the next generation sequencing, so it allows you to do the high throughput analysis. You can study a lot of genes in the same time. And you select some gene from the RNA-seq to do the validation with the nanostring. So the nanostring show the correlate result. We confirm, so it means that we can confirm the result from the RNA-6. And because from the nanostring, you have designed the probe for detection the expression of gene. You can reuse the probe or the code set here for your further study to select some variety of some cultivar of the sugarcane to find the drought-resistant sugarcane variety. Yes, that is quickly about about the concept of the nanostring. So if you have questions, you can ask. Thank you so much. Sorry. Any question?
Would you agree that this appears to be an improved version of microarray? It seems like microarray. The next question is, why would I use this? I would only use this if I want to validate, as per your last slide, if I want to validate some specific genes, then I would use this because it's more sensitive. Two questions.
Actually, I never do the RNA-6. Sorry, I never do the microarray. So I think the microarray, I don't actually know the concept of the microarray, but for this case, we consider to use this one because the nanostring, no need for the step to do the cDNA amplification. So we can directly count the number of the RNA. We consider, why we use this technique, not the microarray. So I'm not sure in the microarray need the application step or not. And I think this so we can reuse the code set to see the expression. Yeah, it's just, I mean that the probe is maybe more specific than the nanostring, I think.
Okay. Yeah. Thank you.
Okay. So the idea behind this then is it's a validating step, right? You get a more accurate measure of the RNA expression change after having gotten a suggestion from RNA-seq that this is something that should be measured. Can you talk about the cost of using nanostring relative to RNA-seq? Is it a substantial additional investment or a small investment?
I would say the nanostring more expensive than the RNA-6. So that is why we have to select some responsive gene for the RNA sequencing first. So in the RNA sequencing, you do the next generation sequencing. So it depends on the number of samples, right? You have more number of samples, it's more expensive. It's more cheaper for each sample. But in case of the nanostring, we do about 48 samples for about 200,000 baht Thai baht. So, but that is that's for the custom process. It will be cheaper for in human commercial courses for an cancer panel something, because for the cancer panel you can do I'm not quite sure, but the number is about 200 genes in the same time. So in this case you can do about 40 genes, about 20 genes plus 4 housekeeping genes. Because in the normalization, you need housekeeping gene. In this case, we use 4 housekeeping gene plus 20 target gene for 200,000 Thai baht. So it's more expensive than the RT-PCR and RNA-seq technique, but it provides a more correct result than another techniques because it's no need for the amplification step.
Okay. Thank you very much. For the US among us, that's something like $6,000, which is a lot, but not insanely large.
And be more cheaper than in Thai because the pro will transfer from the US, I think.
Right. Okay. Thank you very much. Any other questions? All right. Thanks, Ware, for coming over to give us that thought about a new technology for us. Thanks.
Thank you so much.